Hello, viewers. Welcome to today's live on online biology tutorials. I will be reading this chapter number 8 Microbes in Human Welfare, taken from NCERT Class 12th Biology book. I hope my voice is clear. Let me check it out. Yes, the video is also clear and the voice is also clear. So, by now, I hope you must have read this chapter. We will read word by word from NCERT so that you will be able to find it out. Have you left any word, or every word is clear in your mind and you are ready for the boards 2026? So, we are starting with the live reading of Microbes in Human Welfare, chapter number 8, taken from NCERT Class 12 Biology. Besides macroscopic plants and animals, microbes are the major components of biological systems on this earth. You have studied about the diversity of living organisms in class 11. Do you remember which kingdoms among the living organisms contain microorganisms? Which are the ones that are only microscopic? Microbes are present everywhere in soil, water, air, inside our bodies, and that of other animals and plants. They are present even at sites where no other life form could possibly exist, sites such as deep inside the geysers or thermal vents, where the temperature may be as high as 100 degrees Celsius, deep in the soil under the layers of snow several meters thick and in highly acidic environments. Microbes are diverse: protozoa, bacteria, fungi, and microscopic animal and plant viruses, viroids, and also prions that are proteinaceous infectious agents. Some of the microbes are shown to you in figure 8.1 and 8.2. Microbes like bacteria and many fungi can be grown on nutritive media to form colonies that can be seen with the naked eyes. Such cultures are used in studies on microorganisms. In chapter 7, you have read that microbes cause a large number of diseases in human beings. They also cause diseases in animals and plants. But this should not make you think that you would be surprised to know that we use microbes or products derived from them every day. A common example is the production of curd from milk. Microorganisms such as lactobacillus and other commonly called lactic acid bacteria or LAB grow in milk and convert it to curd. During growth, the LAB produce acids that coagulate and partially digest the milk proteins. A small amount of curd added to the fresh milk as inoculum or starter contain millions of LAB, which at suitable temperatures multiply, thus converting milk to curd, which also improves its nutritional quality by increasing vitamin B12. In our stomach too, the LAB play very beneficial role in checking disease causing microbes. The dough which is used for making foods such as dosa and idli is also fermented by bacteria. The puffed up appearance of dough is due to the production of CO2 gas. Can you tell which metabolic pathway is taking place resulting in the formation of CO2? Where do you think the bacteria for these fermentations comes from? Similarly, the dough which is used for making bread is fermented using baker's yeast, which is also known as Saccharomyces cerevisiae. A number of traditional drinks and foods are also made by fermentation by the microbes. Toddy, a traditional drink of some parts of southern India, is made by fermenting sap from palms. Microbes are also used to ferment fish, soybean, and bamboo shoots to make foods. Cheese is one of the oldest food items in which microbes were used. Different varieties of cheese are known by their characteristic texture, flavor, and taste. The specificity coming from the microbes used, for example, the large holes in Swiss cheese are due to the production of a large amount of CO2 by a bacterium named Propionibacterium shermani. The Roquefort cheese are ripened by growing a specific fungi on them, which gives them a particular flavor. Microbes in industrial products. Even in industry, microbes are used to synthesize a number of products valuable to human beings. Beverages and antibiotics are some examples. Production on an industrial scale requires growing microbes in very large vessels called fermenters. Fermented beverages, microbes, especially yeast, have been used from time immemorial for the production of beverages like wine, beer, whiskey, brandy, or rum. For this purpose, the same yeast, Saccharomyces cerevisiae, used for bread making and commonly called brewer's yeast, is used for fermenting malted cereals and fruit juices to produce ethanol. Do you recollect the metabolic reactions which result in the production of ethanol by yeast? Depending on the type of the raw materials used for fermentation and the type of processing, with or without distillation, different types of alcoholic drinks are obtained. Wine and beer are produced without distillation, whereas whiskey, brandy, and rum are produced by distillation of the fermented broth. The photograph of a fermentation plant is shown to you in figure 8.5. Antibiotics. Antibiotics produced by microbes are regarded as one of the most significant discoveries of the 20th century and have greatly contributed towards the welfare of the human society. Anti is a Greek word that means against, and bio means life. Together, they mean against life in the context of disease causing organisms, whereas with reference to human beings, they are pro life and not against. Antibiotics are chemical substances which are produced by some microbes and can kill or retard the growth of other disease causing microbes. You are familiar with the commonly used antibiotic penicillin. Do you know that penicillin was the first antibiotic to be discovered and it was a chance discovery? Alexander Fleming, while working on Staphylococci bacteria, once observed a mold growing in one of his unwashed culture plates around which Staphylococci could not grow. He found out that it was due to the chemical production by the mold and he named it penicillin after the mold Penicillium notatum. However, its full potential as an effective antibiotic was established much later by Ernest Chain and Howard Florey. This antibiotic was extensively used to treat American soldiers wounded in World War II. Fleming, Chain, and Florey were awarded the Nobel Prize in 1945 for this discovery. After penicillin, other antibiotics were also purified from other microbes. Can you name some other antibiotics and find out their sources? Antibiotics have greatly improved our capacity to treat deadly diseases such as plague, whooping cough, which is also known as kali khasi, diphtheria, which is also known as galghotu, and leprosy, which is also known as kusht rog, which used to kill millions all over the globe. Today, we cannot imagine a world without antibiotics, chemicals, enzymes, and other bioactive molecules. Microbes are also used for commercial and industrial production of certain chemicals like organic acids, alcohols, and enzymes. Examples of acid producers are Aspergillus niger, a fungus of citric acid, Acetobacter aceti, a bacterium of acetic acid, Clostridium butylicum, a bacterium of butylic acid, and Lactobacillus, a bacterium of lactic acid. Yeast, Saccharomyces cerevisiae, is used for commercial production of ethanol. Microbes are also used for the production of enzymes. Lipases are used in detergent formulations and are helpful in removing oily stains from the laundry. You must have noticed that bottled fruit juices bought from the market are clear as compared to those made at home. This is because the bottled juices are clarified by the use of pectinases and proteases. Streptokinase, produced by the bacterium Streptococcus and modified by genetic engineering, is used as a clot burster for removing clots from the blood vessels of patients who have undergone myocardial infarction leading to heart attack. Another bioactive molecule is cyclosporin A, that is used as an immunosuppressive agent in organ transplant patients. Is produced by the fungus Trichoderma polysporum. Statins produced by the yeast Monascus purpureus have been commercialized as blood cholesterol lowering agents. It acts by competitively inhibiting the enzyme responsible for synthesis of cholesterol. Microbes in sewage treatment. We know that large quantities of wastewater are generated every day in cities and towns. A major component of this wastewater is human excreta. This municipal wastewater is also called sewage. It contains large amounts of organic matter and microbes, many of which are pathogenic. Have you ever wondered where this huge quantity of sewage or urban wastewater is disposed of daily? This cannot be discharged into natural water bodies like rivers and streams directly. You can understand why. Before disposal, hence, sewage is treated in sewage treatment plants or STPs to make it less polluting. Treatment of wastewater is done by heterotrophic microbes naturally present in the sewage. This treatment is carried out in two stages. Primary treatment. These treatment steps basically involve physical removal of the particles, large and small, from the sewage through filtration and sedimentation. These are removed in stages. Initially, floating debris is removed by sequential filtration. Then the grit, which includes soil and small pebbles, are removed by sedimentation. All solids that settle form the primary sludge and the supernatant forms the effluent. The effluent from the primary settling tank is taken for secondary treatment. Secondary treatment or biological treatment. The primary effluent is passed into large aeration tank where it is constantly agitated mechanically and the air is pumped into it. This allows vigorous growth of useful aerobic microbes like into flocks, masses of bacteria associated with fungal filaments to form mesh-like structures. While growing, these microbes consume the major part of the organic matter in the effluent. This significantly reduces the BOD or biochemical oxygen demand of the effluent. BOD refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by bacteria. The sewage water is treated till the BOD is reduced. The BOD test measures the rate of uptake of oxygen by microorganisms in a sample of water, and thus indirectly BOD is a measure of the organic matter present in the water. The greater the BOD of wastewater, more is its polluting potential. Once the BOD of sewage or wastewater is reduced significantly, the effluent is then passed into a settling tank where the bacterial flocks are allowed to sediment. This sediment is called activated sludge. A small part of the activated sludge is pumped back into the aeration tank to serve as the inoculum. The remaining major part of the sludge is pumped into large tanks called anaerobic sludge digesters. Here, other kinds of bacteria which grow anaerobically digest the bacteria and the fungi in the sludge. During this digestion, bacteria produce a mixture of gases such as methane, hydrogen sulfide, and carbon dioxide. These gases form biogas and can be used as a source of energy as it is inflammable. The effluent from the secondary treatment plant is generally released into natural water bodies like rivers and streams. An aerial view of such a plant is shown in figure 8.7. You can appreciate how microbes play an important role in treating millions of gallons of wastewater every day across the globe. This methodology has been practiced for more than a century now in almost all parts of the world. Till date, no man-made technology has been able to rival the microbial treatment of sewage. You are aware that due to increasing urbanization, sewage is being produced in such larger quantities than even ever before. However, the number of sewage treatment plants has not increased enough to treat such large quantities, so the untreated sewage is often discharged directly into the rivers, leading to their pollution and increase in the waterborne diseases. The Ministry of Environment and Forest has initiated Ganga Action Plan and Yamuna Action Plan to save these major rivers of our country from pollution. Under these plans, it is proposed to build a large number of sewage treatment plants so that only treated sewage may be discharged in the rivers. A visit to a sewage treatment plant situated in any place near you would be very interesting and educating experience. Microbes in production of biogas. Biogas is a mixture of gases containing predominantly methane produced by the microbial activity and which may be used as fuel. You have learned that microbes produce different types of gaseous end products during growth and metabolism. The type of the gas produced depends upon the microbes and the organic substrates they utilize. In the example cited in relation to fermentation of dough, cheese making, and production of beverages, the main gas produced was CO2. However, certain bacteria which grow anaerobically on cellulosic material produce large amount of methane along with CO2 and H2. These bacteria are collectively called methanogens, and one such common bacterium is methanobacterium. These bacteria are commonly found in the anaerobic sludge during sewage treatment. These bacteria are also present in the rumen of cattle. A lot of cellulosic material present in the food of cattle is also present in the rumen. In rumen, these bacteria help in the breakdown of cellulose and play an important role in the nutrition of cattle. Do you think we human beings are able to digest the cellulose present in our foods? Thus, the excreta or the dung of cattle, commonly called gobar, is rich in these bacteria. Dung can be used for generation of biogas, commonly called gobar gas. The biogas plant consists of a concrete tank which is 10 to 15 feet deep in which bio-waste are collected and a slurry of dung is fed. A floating cover is placed over the slurry which keeps on rising as the gas is produced in the tank due to the microbial activity. The biogas plant has an outlet which is connected to the pipe to supply biogas to nearby houses. The spent slurry is removed through the another outlet which may be used as a fertilizer. Cattle dung is available in large quantities in rural areas where cattle are also used for a variety of purposes. So, biogas plants are more often built in rural areas. The biogas thus produced is used for cooking and lighting. The picture of a biogas plant is shown in figure 8.8. The technology of biogas production was developed in India mainly due to the efforts of Indian Agricultural Research Institute IARI and Khadi and Village Industries Commission KYIC. If your school is situated in a village or near a village, it would be very interesting to inquire if there is any biogas plant nearby. Visit the biogas plant and learn more about it from the people who are actually managing it. Microbes as biocontrol agents. Biocontrol refers to the use of biological methods for controlling plant diseases and pests. In modern society, these problems have been tackled increasingly by the use of chemicals, by the use of insecticides and pesticides. These chemicals are toxic and extremely harmful to human beings and animals alike and have been polluting our environment, basically in the soil, groundwater, fruits, vegetables, and crop plants. Our soil is also polluted through our use of weedicides to remove the weeds. Biological control of pests and diseases. In agriculture, there is a method of controlling pests that relies on natural predation rather than introduced chemicals. A key belief of the organic farmer is that biodiversity furthers health. The more variety in a landscape has, the more sustainable it is. The organic farmer therefore works to create a system where the insects that are sometimes called pests are not eradicated but instead are kept at manageable levels by a complex system of checks and balances within a living and vibrant ecosystem. Contrary to the conventional farming practices, which often use chemical methods to kill both useful and harmful life forms indiscriminately, this is holistic approach that seeks to develop an understanding of the webs of interaction between the myriad of organisms that constitute the field fauna and the flora. The organic farmer holds the view that the eradication of the creatures that are often described as pests is not only possible but also undesirable. For without them, the beneficial predatory and parasitic insects which depend upon them as foods or hosts would not be able to survive. Thus, the use of biocontrol measures will greatly reduce our dependence on toxic chemicals and pesticides. An important part of the biological farming approach is to become familiar with the various life forms that inhabit the field, predators as well as the pests, and also their life cycles, patterns of feeding, and the habitats that they prefer. This will help develop appropriate means of biocontrol. The very familiar beetle with red and black markings, the ladybird and the dragonflies, are useful to get rid of aphids and mosquitoes, respectively. An example of microbial biocontrol agent that can be introduced in order to control butterfly caterpillars is the bacteria Bacillus thuringiensis, often written as BT. These are available in sachet as dried spores which are mixed with the water and sprayed onto vulnerable plants such as brassicas and fruit trees, where these are eaten by the insect larva. In the gut of the larva, the toxin is released and the larva gets killed. The bacterial disease which kill the caterpillars but leave other insects unharmed because of the development of methods of genetic engineering in the last decade or so. The scientists have introduced Bacillus thuringiensis toxin genes in two plants. Such plants are resistant to attack by insect pests. BT cotton is one such example which is being cultivated in some states of our country. You will learn more about this in chapter 10. A biological control being developed for use in the treatment of plant disease is the fungus Trichoderma. Trichoderma species are free living fungi that are very common in the root ecosystems. They are effective biocontrol agents of several plant pathogens. Baculoviruses are pathogens that attack insects and other arthropods. The majority of baculoviruses used as biological control agents are in the genus Nucleopolyhedroviruses. These viruses are excellent candidates for species specific narrow spectrum insecticidal applications. They have been shown to have no negative impact on plants, mammals, birds, fish, and even on non target insects. This is especially desirable when beneficial insects are being conserved to aid in an overall integrated pest management or IPM program or when an ecologically sensitive area is being treated. Last topic, microbes as biofertilizers. With our present day lifestyles, environmental pollution is a major cause of concern. The use of chemical fertilizers to meet the ever increasing demand of agricultural produce has contributed significantly to this pollution. Of course, we have now realized that these are problems associated with the overuse of chemical fertilizers, and there is a large pressure to switch to organic farming, the use of biofertilizers. Biofertilizers are organisms that enrich the nutrient quality of the soil. The main sources of biofertilizers are bacteria, fungi, and cyanobacteria. You have studied about the nodules on the roots of leguminous plants formed by the symbiotic association of rhizobium. These bacteria fix atmospheric nitrogen into organic forms, which is used by the plant as nutrient. Other bacteria can fix atmospheric nitrogen while free living in the soil. Examples Azospirillum and Azotobacter, thus enriching the nitrogen content of the soil. Fungi are also known to form symbiotic associations with plants, mycorrhiza. Many members of the genus Glomus form mycorrhiza. The fungal symbiont in these associations absorbs phosphorus from soil and passes it to the plants. Plants having such associations show other benefits also, such as resistance to root-borne pathogens, tolerance to salinity and drought, and an overall increase in the plant growth and development. Can you tell what advantage the fungus derives from this association? Cyanobacteria are autotrophic microbes widely distributed in aquatic and terrestrial environments, many of which can fix atmospheric nitrogen. Anabena, nostoc, oscillatoria, etc. In paddy fields, cyanobacteria serve as an important biofertilizer. Blue-green algae also add organic matter to the soil and increase its fertility. Currently, in our country, a number of biofertilizers are available commercially in the market, and farmers use these regularly in their fields to replenish soil nutrients and to reduce dependence on chemical fertilizers. So, with this, I am done with the reading of microbes in human welfare. If it was helpful to you somehow, if it helped you in pronouncing the scientific terms clearly, then do hit the like button and share it with your friends so that they can also revise the chapter in this unique way. Once you are done with your chapter understanding, always listen to these audios. They help you in grasping. Have you understood all the terms that are given in the chapter, or still some more understanding is required? Stay connected for more such learnable videos. Thank you for joining today.